Namaskara, namaskara, namaskara. So in this video, we will discuss what is Pilli and Fimbria. We have already discussed, that bacteria have different kind of appendages, and in that all appendages only, these are another kind of appendage, and that is called pilia. Sorry, that is called Pilli and Fimbria. Fine. What is this Pilli and Fimbria? There is not much difference between Pilli and Fimbria. Both are surface appendages. Only thing is that this Pilli is the longer in size and Fimbria is smaller in size. Yeah, the Pilli size wise you will see. So if we have a bacteria, so Pilli will be long. Yes, Pilli will be long. Okay, and our Fimbria is shorter in size. Okay, either it is Pilli or it is Fimbria. Both are surface appendages present in the bacteria, and they are thin, and they made up of protein tubes which are originated from the cytoplasm of the bacteria. You see, like we have one bacteria here. Can you see here? We have some bacteria here, and this bacteria, these all longer structures are Pilli, and the smaller structures are actually bacteria. The smaller structures are Fimbria. Fine. Pilli, if we'll compare, Pilli is typically longer, right? And our Fimbria is comparatively smaller in size. Pilli, is present in gram-negative bacteria, but this Fimbria, is present in both gram-positive and gram-negative bacteria. Yeah, Fimbria is present in both gram-positive and gram-negative bacteria. Pilli used for bacterial conjugation. Yeah, what is bacterial conjugation? We will see in detail. Bacterial conjugation is something, when the two bacteria will attach with each other, and then they will transfer genetic material. That is only bacterial conjugation. We will see in detail. So Pilli, it's used for bacterial conjugation, and this Fimbria, is mainly used for attachment to the surface. See, attachment to the surface, both have basic function is attachment to the surface only. But in particular function, if you'll see, then Pilli is used for bacterial conjugation, and the Fimbria is used for cell attachment to the surface. Pilli is comparatively less in number. In one bacteria, I'll see generally they are 1 to 10 in number, right? But Fimbria, the Fimbria will be 100. They'll be like several hundred in number. Yeah, you see, these all smaller structures are Fimbria. Fine. Now we'll see the types of Pilli. Yeah. So if you'll see types of Pilli, one second. What happened? Yes. Okay, fine. So when you will see the types of Pilli, Pilli are two types. One is short attachment Pilli, and another one is the long attachment Pilli. Right? See, either it is Pilli or Fimbria, they have a hair-like structure, right? That is called shaft. But at the end of that, at the tip of this hair-like structure, they have some kind of glycoproteins present here, or glycolipid present here, which act as a attachment site to binding to the host. Like suppose they have to bind to any kind of host cell. This bacteria have to bind to any kind of host cell, so they will bind it by the help of this structure which present at the tip of their Fimbria or Pilli, and the structure is made by glycoprotein or glycolipid. Okay, it will bind to the receptor of the host cell, and the things will be done. Now this this Pilli have two type. One is short attachment Pilli, another one is long attachment Pilli. Short attachment Pilli is the short and is numerous actually, and they will attach to the surface. Yes, they will attach to the surface, and they will resist in flushing. Yes, they will resist it flushing, like water is flowing there. Okay, so they will resist to that flushing activity of the water. Another one is the long conjugation pili. This long conjugation pili is also called sex pili or F pili, right? They have a super important role, in the transfer of genetic material from one cell to the another cell. Like, try to understand it. We have one bacteria. One second. Like, you see, here we have two bacteria. This is bacteria A and this is bacteria B. Bacteria A want to transfer its genetic material. Suppose it have some kind of extra genetic material and it want to transfer this genetic material to bacteria B. So it will attach with the is the F pili, and they will form one tube like structure. This tube like structure is called conjugation tube. Okay, and via this conjugation tube they will replicate their genetic material and they will transfer one another copy to this another bacteria, right? So gram negative bacteria is typically transferring DNA from donor to the another bacteria via sex pillars, right? Or by that long kind of pili to the another bacteria, right? And this process is only called conjugation. The transfer of genetic material via this kind of conjugation tube is called it's called uh, conjugation, and this conjugation tube is formed by F pili, right? And not only that, this pili, is also causes formation of antibodies against it. Yeah, they act as a antigen also. Antigen, antigen is substance which causes production of antibodies. So if they will enter in body, possibly they can cause formation of antibodies against them also, right? They have epitope. Actually, on that epitope the bacterial antibodies will come and they will bind there. Fine. So in this video what we discuss, students? In this video we discuss what are the in this video we discuss actually what are the fimbria, and we discuss what are the we discuss what are the pili, and then we discuss what is difference between pili and fimbria, and then we see what are their significance. Remaining we'll see in next video.